Chase bacteriophage experiment. This is the early experiment, and we will discuss this experiment in this lecture. Tobacco mosaic virus experiment, the structure of the DNA inside the nucleosome, the Watson and Crick model of DNA, the double helix, organization of DNA or RNA in the chromosome. What is the nature of the genetic material? It's a question scientists try to answer. It's a stable source of information. It has ability to replicate accurately. It can be changed. It's stable, replicate, it change. There is a timeline for events. In 1890, Weizmann substance, the nucleic, the genetic material, a substance in a cell, nuclear controlled development. Weizmann. Genetic material is a substance in the cell nuclei controlled development. In 1900, chromosome shown to contain hereditary information. Later shown to be composed of protein and nucleic acid. In 1928, Griffith transformation experiment. In 1944, Avery transformation experiment. In 1953, Hershey-Chase bacteriophage experiment. In 1953, Watson and Crick proposed double helix model of DNA. In 1956, Yearer and his colleague demonstrate RNA is a viral genetic material in tobacco mosaic virus. This is the experiment of Frederick Griffith. Transformation principle demonstrate with Streptococcus pneumoniae. Streptococcus pneumoniae is a bacteria, a pathogenic bacteria. Frederick Griffith transformation experiment conducted in 1928 was a series of experiments on Streptococcus pneumoniae that lead to the discovery of the bacterial transformation, a process by which bacteria take up foreign genetic material. In this experiment, Griffith used two strains of S. pneumoniae, a virulent strain that caused pneumonia and a non-virulent strain that didn't cause pneumonia. In one experiment, he injected a mice with S strain, which caused death. In another experiment, he injected mice with the R strain, the rough strain, which didn't cause death. In the third experiment, heat killed the S strain, making it non-viable, and injected it in mice, which didn't cause death. And heat killed the non-virulent, survived, no bacteria recovered. Here, he combined both the heat-killed non-virulent and the living non-virulent, and inject the mice, and the mice died, and the virulent bacteria. Here, the Oswald Avery transformation experiment, and determined DNA is a genetic material. Oswald and Avery transformation experiment in 1944 demonstrated that DNA was a genetic material responsible for the transformation of non-virulent bacteria into virulent bacteria. Prior to this experiment, it was believed that protein were the genetic material. Avery, along with his colleague Colin MacLeod and McCarthy, purified various cellular components of the Streptococcus pneumoniae and tested their ability to transform non-virulent bacteria into virulent bacteria. Treat with the RNAs, add DNA to our bacteria, blade growth, S transformant produced, mixture of DNA and RNA. Here a mixture of DNA and RNA, we treat it with DNAs, add RNA to our bacteria, blade on growth media, no S transformant appear. Here S transformant produced. Hershey-Chase bacteriophage experiment in 1953. The bacteriophage, as we know, a virus that attack bacteria and replicate by invading a living cell and using the cell molecular machinery, hacking the molecular machinery of the cell. Hershey and Chase experiment conducted in 1952 by Alfred Hershey and Martha Chase was a series of experiments that helped to confirm that DNA is the genetic material in cells. The researcher used bacteriophage, which are viruses that infect the bacteria. To investigate how genetic material is transmitted, they labeled the DNA in the phage with radioactive isotope of phosphorus 32 and the protein code with the different radioactive isotope of the sulfur 35. They then allowed the labeled phage to infect the bacteria and examine which radioactive marker ended up inside the bacterial cell. In the life cycle of the phage, it entered the bacteria and breakdown of the bacterial chromosome totally broken down, and the phage chromosome start to replicate, and the expression of phage genes to produce structural component, assembly of the progeny, and release of progeny phage. Here the bacteriophage is composed of DNA and protein set up to replicate, label DNA with phosphorus 32 and label protein with sulfur 35. Infect E. coli bacteria with two types of labeled T2. B32 is discovered within the bacteria and the progeny phage, whereas S35 is not found within the bacteria but released with the phage go. Here, shrimp tobacco mosaic virus experiment in 1953. The viral strain to demonstrate RNA is the genetic material of tobacco mosaic virus. RNA also is a genetic material in some viruses, and this is was an experiment. Gare and Schramm in 1956. He is a German. Experiment on tobacco mosaic virus demonstrated that the viral RNA alone is sufficient to induce viral infection in host cell. They extract and purified the RNA component from tobacco mosaic virus and found that it was still able to infect and replicate within the host cell. This was a pivotal experiment that helped establish RNA as a genetic material. And he gave a lecture in Max Planck Institute, explain his experiment. For conclude these experiments, the early experiment, Griffith and Avery, DNA not RNA is transforming agent. Hershey and Chase, DNA not protein is the genetic material. Gare and Schramm, RNA not protein is genetic